Okay. First, thank you. Sorry, apologize for not being there in person due to both weather and a plane running off the runway here in Montreal. So it became impossible there. But again, I thank the organizers of the meeting for allowing me the opportunity to present some of the work which was sponsored by the HCU. So, without further ado, I'm going to speak a little bit about our attempt to look at aminoglycosides as a treatment for inborn errors in vitamin B12 metabolism. So, to begin with, as a conflict, I have received some support in the past from Recordati, which is a company which you may be familiar with. So, as a sort of an overview of the work, I just want to remind everybody that vitamin B12 metabolism involves actually, and its utilization requires a number of genes, which the laboratory I'm part of has been involved in actually delineating and identifying. So you can see, which I have not actually included in this particular slide, it comes from the GI tract, it enters the cell by a particular transporter, ends up in the lysozyme. There are two proteins involved in its export. The names cobalamin J and cobalamin F would actually refer to complementation groups, which reflects a method how these genes are actually identified initially. So one of the first steps, once cobalamin, whatever form is absorbed, is actually handed over to a protein called cobalamin C. Cobalamin C is actually a protein which is involved in channeling the vitamin B12 to different pathways, and which doesn't actually kind of reflect in the slide. Actually, cobalamin C converts basically all forms of cobalamin, whether it's cyano or even methyl or anything that's attached to one with basically almost just a hydroxy group there. And it actually forms probably a complex, or at least interacts with cobalamin D, which is involved in channeling some cobalamin into the mitochondria, or more to focus today is forming an interaction with cobalamin G, or methionine synthase, as it's called. And actually, probably cobalamin C actually probably forms a complex with cobalamin G, or methionine synthase, which is a focus of today, and with cobalamin E, which is involved in maintaining methionine synthase, MTR, in a functional status. So really what cobalamin G, or methionine synthase, or MTR, whatever, these are all synonymous days, is actually involved in transferring the methyl group from methyl tetrahydrofolate to basically homocysteine to regenerate methionine. That's its key role that's identified. And knowing this actually has implications in terms of what can be done to treat people with defects in this disorder. So this is probably an actual complex between cobalamin G and cobalamin E, probably cobalamin D and cobalamin C. Then there's some information that has actually been shown to do that. They form, if not an actual complex, at least an interactor. Just to sort of focus a little bit more on cobalamin G, what cobalamin G is actually doing is taking, as we said, is taking methyl tetrahydrofolate, transferring the methyl group. It forms this methylcobalamin intermediate, which is obviously bound to MTR, methionine synthase, transferring the methyl group to homocysteine, regenerating homocysteine, and this pathway keeps reoccurring. As it turns out, the cobalamin molecule to which actually the methyl group is actually ligated to, it actually sometimes undergoes a spontaneous oxidation and has to be reduced by another enzyme called MTRR, or cobalamin E. So this is actually an important enzyme in maintaining the functionality of cobalamin G. And it turns out part of this role is MTR is actually catalyzing the transfer of the proper a methyl group from adenylyl methionine to generate adenylyl homocysteine. You'll see this again to regenerate and, if you will, prime the pump here. So it's actually probably a two-stage reaction that this MTR is actually catalyzing. And, if you will, not only maintaining it, probably primes the pump to get methionine synthase functioning. So let's look a little bit more closely at methionine synthase. Sometimes I call it MTR, which is the gene product. Sometimes I call it cobalamin G, which is sort of the complementation group, which is all referring to this particular enzyme, which is converting methyl tetrahydrofolate and homocysteine to methionine. So cobalamin G, MTR, when there's a defect in this enzyme, particularly it produces a disease, which an autosomal recessive disease that's affecting this enzyme. This is actually the gene name. The proper name is a little bit larger. You can see it reflects what it's doing. Cobalamin vitamin B12 dependent enzyme 5-methyl tetrahydrofolate homocysteine methyltransferase, which is again is transferring the methyl group from methyl tetrahydrofolate to homocysteine, and methionine synthase name. And it's a rare disorder. Only about 70 individuals have been described in the literature. And unfortunately, people who suffer from this disorder, multiple systems are affected. They can have, in some cases, failure to thrive. They can have blood and bone marrow defects, various nervous system defects. A smaller percentage may have eye disorders, and even a smaller percentage yet may have renal disorders. Again, with sort of an HUS type phenomenon. So it affects multiple systems for people who have a defect in this particular gene. So recently, just to put it in perspective, Dr. Watkins in our laboratories actually was a keynote. He actually assembled put together the genes, the mutations or variants that have occurred in MTR that have been described in the literature and within our laboratory. And as you can see in the next slide, there's a number have been described in the literature. Some of them actually have just been defined within the laboratory in a relatively recent publication. So you can see if this is the structure of MTR, you can see it's broken up into a number of coding regions or exons, over 33, and each of these regions perhaps have different have functions. One's towards the front of the gene, maybe involved in homocysteine binding, some in methyl tetrahydrofolate binding, some in binding vitamin B, cobalamin binding, that is vitamin B12, and some in binding S-amylose methionine. And in fact, just as an aside here, you can actually classify the different variants. Some are splice variants, some are deep intronic variants, probably affecting expression. Some are insertion deletions. Many of them, 36 of the number of mutations of the 80 mutations described in this patient are missense. That will put an incorrect amino acid. In fact, at this very end, this is actually here, this mutation P1173L is actually one of the more common mutations or variants you find in this gene, and this is actually functioning in the SAM binding region. A certain percentage, which is the point of our discussion today, some are called stop mutations or stop variants, and about 8% of all these mutations that are variants that we described are actually stop mutations, and these I have indicated by little red stars here. Those are all stop mutations. At least half of them are in the SAM binding region, and you'll have a few scattered in other regions. And this is the focus of the day. So basically, you'll see there are, as you can see from this overview of where you find mutations, that there's a variety of mutations. Some are slightly more frequent than others. I said that P1173L is probably the most frequent one, but a certain percentage are stop mutations, 8%. And actually, if you look at, generally speaking, amongst other genes, some genes will tend to have about 10 to 20% of all mutations, and other genes will also be stop mutations. So our focus as the work is to look at what we can do in particular classes of mutations, what we call stop mutations. mutations, which I'll talk about a little bit and help define. So just as a reminder, when we get patient-derived fibroblasts as part of the diagnosis, there's actually two assays that we can do on the patients within the laboratory. One is historically what's called propionate incorporation, where you take radioactive propionate, and this actually can be used to actually look at sort of all functions that are associated with cobalamin C and earlier, and those that are involved in the mitochondrial pathway, which is involved in adenosylcobalamin. So that's what you're looking at when you're looking at propionate incorporation. More specific to our talk today, we're going to look at methyl tetrahydrofolate incorporation, where we're actually looking at, if you will, the function of methionine synthase for all intents and purposes. So basically, you're giving radioactive methyl tetrahydrofolate, you're feeding fibroblasts derived from patients in the presence of homocysteine, it's incorporated into methionine, and eventually will end up into proteins and other materials, which we can actually quantitate. And this is the assay we're actually using, as you'll see in the work. So we're actually looking more specifically at the methylcobalamin pathway and cobalamin G function. So this is just a compendium of all the methionine synthase mutants that we actually have in a measured lab. You can see it's been over the years accumulated about 47 different lines. These actually represent a variety of mutations in particular. And you could actually look at the propionate incorporation just to give you an idea. So these individuals, the propionate is affected, so it's about 8.4 nanomoles per milligram protein. Control would give about 16 a wild type. So there is some effect at that. But what we often use to look at some of these lines, these are all cobalamin G lines, is if they're stimulated by adding exogenous cobalamin in the form of hydroxycobalamin, and these don't show any stimulation. Certain mutations in other mutations in other complementation groups will be stimulated by cobalamin, but not the cobalamin G lines at this particular. More to the point, when you actually look at methyl tetrahydrofolate incorporation, they'll have a value, again, of about 32, and the control being 100 of units, if you will. And again, these do not show any stimulation with hydroxycobalamin, not effective. I've just highlighted one particular line, which is I call patient 9. This patient 9 is patient 9 in the paper, and this patient actually is the one carries heterozygous for a stop mutation. And again, here, just like the other lines, it does not. It shows reduced propionate incorporation, clearly reduced propionate incorporation, and no stimulation with hydroxycobalamin. You might see a difference in amount of incorporation in this particular report compared to some of the other slides at that point, because we've altered the assay slightly to get more counts into an increased exposure time. So sometimes the numbers aren't exactly the same as when this patient was originally identified a number of years ago, but it shows the same pattern. The next 3 slides, I'm just going to very briefly look at. I'm only going to just mention them because they represent sort of both unpublished observations by one of our graduate students and also a report by from Dr. Banerjee's group. And this is the characteristics, if you will, of how much of a particular substrate is needed to get sort of maximal activity of this particular product, cobalamin G. And actually, you need actually quite a bit of homocysteine in the wild type to actually get sort of half maximal activity. You get about 3 micromoles per liter. Methyl tetrahydrofolate, you need about 100 micromoles. And this is actually what Ms. Metabolic, who you'll see later on, is actually can corroborate this with looking at fibroblasts as opposed to purified enzyme. And again, SAM, you don't assess them on methionine, you still low concentration, those are nanomoles per liter. This is just one, that very common variant. You can see sometimes there can be effects on enzyme characteristics of these particular substrates. Here, SAM loading, you'll notice it went from 190 to 8 micromoles. So that's about close to that's about 1,000-fold difference. So sometimes it'll affect the actual particular region of binding. These two slides, I wasn't too sure myself whether to go over them or not, but it does, and maybe it may be more suitable for the question and answer, but some of this basic work, both in vitro and in vivo, reflects on what is sort of the conventional pathway of treatments of these particular individuals. So individuals with cobalamin G, they'll often give them oral folate. It could be in the form of methyl tetrahydrofolate or folinic acid. It's not clear what concentration should actually be given, because when you actually look, for example, at the Km, the amount of substrate that's needed to get the sort of half maximum activity for methyl tetrahydrofolate, it's around 100 micromoles per liter, and normal reference range is 7 to 40 nanomoles per liter. And when you give patients folate, you're not be able to increase that very much. And it's unlikely giving the patient 1 to 5 micrograms per day will not achieve this. What I'm questioning is that depends your objective. If you're trying to drive the enzyme forward on its own, you need a lot of methyl tetrahydrofolate. I'm not sure it's achievable. Clearly, without going into details, you want to provide folate for other steps, and that's why you give them folinic acid. But how much you're giving them, that's something that's a little discussion. Homocysteine, now this is counterintuitive. Homocysteine is required to drive the reaction forward, but you have to be careful about allowing homocysteine to rise, because that's associated with thrombotic risk. And in fact, both values that we see only get homocysteine saturation around 100 micromoles per liter, which is paid for people who will be treating for homocystinuria. I'm sure will say, well, that's a large amount. That's a large amount, and usually they try and get them down to 50 micromoles or less. And actually, what we see in vitro studies and not published work, this provides a reasonable amount of activity of wild type methyl tetrahydrofolate of methionine. I'm going to not mention dihydrolipoic acid, because that's some of the based on the observations of Dr. Banerjee's group that may be useful. Usually you give the patient parental hydroxycobalamin. This is sometimes responses implicit stimulation data. What concentration is required to saturate the system? Well, probably it's fairly high. And how you're giving it? Probably best hydroxycobalamin, because what happens is cobalamin C will take methylcobalamin and convert it to hydroxycobalamin or another cobalamin bound derivative. Methionine, well, you usually want to give a sufficient amount to make sure that you have enough amount of growth, but straight enzymologies are producing the back reaction, and it's not even clear when you're giving oral methionine what pools. There are separate pools being generated. Again, these are unanswered questions that have to be added. Ethinyl. Some people say, why not give the patient SAM, ethinyl-methionine? Well, the problem is, and to try and circumvent methionine deficiencies or to improve MTR reactivation. The trouble is, as far as we can understand, SAM isn't actually transported across the cell membrane. You can buy it as a dietary supplement. It's not sure that it ever gets into the cell. Some people will say, well, maybe you can replenish SAM with methyl thioadenosine, which is sort of a metabolite of SAM, but it's actually that dependent on methionine synthase function to regenerate SAM. So the questions of utility of SAM or methyl thioadenosine are really up in the air. And of course, MTR reactivation, that's an FAD-dependent enzyme, so FAD makes sense. I guess I just want to mention this, is that sometimes we have to think carefully and clearly what we're doing in terms of replenishment in sort of the standard pathway. Is there something better? That sometimes will require either clinical studies, but sometimes we can be guided by what occurs in vitro with purified enzymes or what has occurred in the fibroblast cell lines. So with that sort of digression and trying to respect my time, so let's look at the actual work. It's trying to look at variants in methionine synthase, specifically stop mutations. So you have to realize just that when the genome contains the specification of the genes, it produces a messenger RNA, which sort of transfers information from the DNA to the protein translation machinery. The genes actually have punctuation that says a gene starts here and ends here, and where it ends is called a nonsense mutation. And when these occur, when a regular base is changed to one of these stop mutations, it'll say, oh, we'll stop translating the protein at that point in time. Sorry, I just wanted to. Okay, thank you. So just wanted to make sure I don't overstep my boundaries of my time. So basically, you're adjusting the punctuation, saying a protein will start here and end here, and if you have a regular sense mutation or one that specifies an amino acid being converted to one that says stop, you may have a short truncated product which is non-functional. So we're decided to concentrate on these one particular class of mutations. This was basically inspired by some collaboration that I was involved with looking at another disease called cystinosis. We're actually using a specific novel aminoglycoside, which you'll hear about later, to allow translational read-through and allow return functionality to the gene product in this particular disease called cystinosis. We're going to be looking at one particular class of pathogenic variants, which are called stop mutations. You'll see them sometimes called premature termination codons. Now, for people who read the literature or even the press, sometimes you can say, what is the availability of any procedure that we develop in the lab? So we decided we'd concentrate on low molecular weight compounds, or small compounds that are available, that may have undergone or undergoing clinical trials. So that affects the availability if they prove useful. And these do not require a delivery vehicle, so they don't have to be transported. Because they're small, they can sometimes be taken orally, and they could be absorbed. So we want to look at a specific class, looking at availability. Now, unlike others, always in every case where people looked at these compounds that allow to overcome these stop mutations or read through these stop mutations, we wanted to make sure that they were relevant to this disease. So we looked at our experimental vehicle were patient-derived fibroblasts, not animal models, nor plasmids for in vitro expression. We wanted to see patient-derived fibroblasts. So that's as close as you could get to experimenting with the actual person. We also wanted to look at some function that was directly relevant to the disorder. In this case, methyl tetrahydrofolate incorporation which reflects methionine synthase or cobalamin G function. So we want to be as close as possible to looking at a situation of clinical relevance. So again, we're going to, as we saw, we were going to look at the methionine methyl tetrahydrofolate incorporation. So this is just sort of reviewing what we mean by an agent to overcome a stop mutation or a premature termination codon. So remind us that DNA is transcribed into mRNA, which is the process called transcription. They link individual nucleotides to get an mRNA product. The mRNA then has to be translated. By that, we mean you have to link amino acids together to produce the functional protein. So the machinery that does, if you will, the translation is called the ribosome. So the ribosome is a structure, you may remember your biology, it binds the mRNA, and it actually transfers using from tRNA, which is the DAP, which is sort of carrying an amino acid, it transfers that to your growing polypeptide chain. And this chain will keep going until you come to a stop codon, which occurs naturally. And at that point in time, various release factors will bind to the structure and cause release of the polypeptide chain. So you come, this is a typical stop codon, UGA. That's sort of a trinucleotide, a trinucleotide codon. So this is normally what's happening. The problem is where if a mutation that produces a stop codon prematurely, you'll get a small product, a non-functional product, which does occur. So what people have noticed in the past, that certain antibiotics, primarily gentamicin, will actually alter the binding, will actually bind to the ribosome, and instead, when you come to a particular stop site, it will actually allow the binding of an amino acyl tRNA, a tRNA containing an amino acid. And this will actually will read through and will actually end up transferring an amino acid to the polypeptide chain. Now, the actual incorporation can be random, what gets incorporated. The main point is an amino acid is incorporated, allowing the continuation of elongation of the chain, if you will. So you're getting, in the terminology, a non-cognate tRNA binding and incorporating an mRNA. So this drug is allowing, if you will, the misreading of a stop codon as a sense codon or a codon of another protein, of another amino acid. So that's the basic idea. So people have actually looked at a number of agents to try and allow read-through to get through these stop mutations. So we've been looking at agents as different categories. We've looked particularly those that allow PTC read through these stop codons. As you'll see, we looked at adalurin, gentamicin, well, actually gentamicin analog, and a compound called ELOX-O2, which is a non-toxic. And the reason people don't use gentamicin or genticin, as you'll see, is it's fairly toxic, causes both kidney toxicity and autotoxicity here. So you can't use that compound directly. So you want to necessarily use other compounds. We did look at two other compounds, which I'm not going to talk about. We looked at clitosine, which actually affects translation, and we also looked at diaminopurine, which actually affects another step. It's actually kind of an adenosine analog, if you will. But here it is, it allows PTC read-through, but we found, which I'm not going to talk about very much, that they're actually, diaminopurine and the clitosine are actually too toxic. They're just not useful, and they did not display an activity. So the compounds we look to screen, if you will, genticin, which is sort of the one that most people look through most directly. It's related to gentamicin. It's only used investigational use. Its use would be limited in allowing read-through by auto and nephrotoxicity. But a group in Israel developed a drug which they call ELX-O2 or adalurin, which also targets eukaryotic ribosomes, but it's actually almost non-toxic. And if you will, and it's actually undergoing clinical trials. So again, we're trying to utilize some things that are available. We looked at adalurin, which was produced by a company a number of years, PTC Therapeutics, which actually was marketed for a while for treating stop mutations in Duchenne's muscular dystrophy. So it is controversy how effective it actually was in treating that disorder. So it's no longer used for that, but it was approved and had gone through some clinical trials. Clitosine, which I mentioned, was investigational use, and that's an adenosine analog, and diaminopurine, it's an adenine analog. They're really for investigational use at this point in time. Moreover, as I mentioned in our hands, clitosine diaminopurine turned out to be toxic. So let's look a little bit better at the stop variants that we have in MTR. About 6 out of 80 of the identified mutations are stop mutations at this point. Many of them actually turned out to be in our biobank at that point in time. And the one where we'll actually concentrate, we use as sort of our keynote for our studies was so-called patient 9, who's in our biobank and is heterozygous for a stop mutation. It's an arginine that gets replaced 1147 and also has a splice mutation in exon. And we also know some others that have been identified of slightly different structures that have been available. These actually, some carry the same stop mutation and some have others. Unfortunately, we don't have a homozygous, so we have a heterozygous ones. That means for us to have maximal effect that we'd see in turning an affected fibroblast line to a wild type line, we'd see maybe only 50%. That would be our maximum. And as we can study, what we looked for is a minimum for any line carrier mutation. We want at least a 2-fold increase over baseline activity to suggest a compound might be clinically useful. The reason for that is even all these cell lines that we have with stop mutations do have some baseline activity at this point in time. So we want to make sure it's a clean delineation. So I'm not showing you that we established dose response curves. We try to choose the dose of each drug that's optimum in terms of effectiveness and toxicity. We found that exalurin and atalurin are basically not toxic, exalurin being a gentamicin derivative that's been selected for low toxicity. Atalurin was one produced by a PTC company. Gentamicin is not used clinically because it has some toxicity, and clintosine and 2,6-aminodipurine are highly toxic to cultured fibroblasts. So they're unlikely to be useful. So let's just look through the actual results. So here's gentamicin. So here we're going to look at a control cell line with or without hydroxycobalamin, our patient cell line which is heterozygous for a stop mutation, plus or minus hydroxycobalamin. So I just point out the circles are all those that are being treated with the drug. The circles represent always those that are treated with the particular drug, and the diamonds are those that before drug treatment. So even in the controls with gentamicin, you'll see this level of incorporation, about 500 units. You particularly add the drug. There is a diminution of methyl tetrahydrofolate incorporation, a measure of methionine synthase function. These represent the mean of 2 separate experiments. We want to repeat the experiments to obviously show consistency. When we looked at gentamicin in particular, our indicator cell line, basically gentamicin didn't do anything. And this just actually puts relative values, just sort of in a different way. So looking at control cell line, adding gentamicin actually decreased incorporation. And you can see the patient 9 cell line, the one heterozygous stop mutation, also some diminution, but there's really no stimulation with the drug. So this is not allowing effective read-through. We looked at exalurin, which is a non-toxic aminoglycoside. Again, the circles represent those with treatment, diamonds prior to treatment, and again, you can see that the exalurin is actually not really, not toxic at all compared to what we saw in the previous experiment. Again, no effect on the patients. the patient heterozygous for the stop mutation. And this is just showing this graphically. This is just showing a little more indication. Basically, as a relative measure, no effect, basically. Xylurin with or with hydroxycobalamin, no effect. So let's look at altelurin. So this is again a compound produced by PTC Corporation. It was tried and had a limited trial with Duchenne muscular dystrophy. So we actually repeated this experiment a number of times to actually see what is occurring to make sure that it's real at that point in time. And you'll see why in a minute. So you can look at different replicates. Sometimes experiments will vary a little bit from time to time. So circles with treatment, diamonds without treatment. And basically, you can see really no effect or incorporation in our control cell line. But what we could see consistently over 3 experiments, we repeated this, that actually altelurin, especially with hydroxycobalamin, did produce a slight stimulation in incorporation, in methylene tetrabible incorporation. Again, we're looking, remember, we were looking, if the baselines are 100, we're looking to really know about incorporation around 200 to be a clear benchmark that it might be clinically useful. So again, our control cell lines, no effect, altelurin plus minus hydroxycobalamin, this being like 100% incorporation. And if we look at fibroblast 9, a heterozygous one, here that learned it actually did produce some stimulation, and you can get about a 26% increase over baseline with altelurin and hydroxycobalamin. So there is a consistent stimulation of incorporation adding this drug, which does produce read-through. But looking at all this, our actual final conclusion was that read-through does work, but in a very limited fashion. It does produce a proof of concept. That altelurin is synergistic with hydroxycobalamin, but our response was very low. At best, it's about 26%. And this, in our mind, we did not feel this is really ultimately clinically useful. Remember, our benchmark was we were looking for about a 2-fold increase over baseline to suggest a compound may be clinically useful at that point. So it doesn't produce very much, it does produce a response, a reproducible response in a very limited fashion. So before this idea of read through the works, we'd need a significant increase in efficiency. We can talk about reasons why these drugs, these particular drugs may have limited efficiency, but an increase in efficiency would be required. So our thinking is to look at stop mutations. Probably the more effective way is to use anticodon engineered tRNAs. These are tRNAs which you synthesize that actually have that are specific for a read-through, and they can insert the correct amino acid rather than a random amino acid, but putting in the correct amino acid. And this is where our thinking is going in terms of trying to overcome stop mutations in an effective fashion. So just to wrap up and not to exceed my time, I would just thank HCU Network for their support. It was great working with you. And this is our team who's worked. That's myself, Dr. Rosenblatt. This is Caitlin with the little red star who actually did the work you see. This is two of our graduate students, Nagar and Ragged, who's actually done work who did the work, who's actually looking at methylene tetrahydrofolate incorporation and some summer students. We were out for lunch. So with that, I will thank very much HCU, and that's the end of the presentation. Thank you very much. I hope I didn't exceed my time.
Yes, this is Leo Dielman from the Netherlands. I was just wondering whether the increase in enzyme activity by 26% was actually associated with indeed a longer, high percentage of full-length mRNA for the enzyme.
I think the question is, what is the mechanism of alpha-leurin producing? I mean, is it producing a larger number of full-length mRNAs? Is that the question?
Yes, that's the question. Thank you.
We don't know. We weren't able to do southern blotting, etc., or equivalent to look at actually the size, or do any qPCR to actually look at the size of the mRNA product at that point in time. So that I can't quite answer, but it seems to be, I mean, there's been a PNAS articles in the last 1 or 2 years, which seems to show it does allow and read through, but some people actually consider, what can I say, combining inhibitors of nonsense-mediated decay and looking at that particularly in concert with some of these materials, and maybe that will enhance the action. But again, specifically, is as opposed to pushing a non-cognate amino acid in or increasing the amount of availability of mRNA, I can't formally address, say, yes or no to